And the Great Paper Mache project is still going on, at least in my house it is. And so let's have a look at, firstly, methods, and then we'll have a look at what you need to do for the rubric. So remember, last week you should have had 2, 3, and 4 finished, and this week it's 4 to 7, I think. There's 4 of them this week, and that should be due very soon. So let's have a look what you need to do, because once you've done that first bit that you've already done, the rest of it is a whole lot easier. The only sticking point is when you look at the gene affected and the protein modified, which is why I made a big fuss about that before the school was closed. So let's have a look at, basically, you've seen this before. This is my pelvis of a Rudopithecus, an ape ancestor of humans. And the importance of this is, of course, it's different in several important ways from that of the great apes. And people used to think that humans were descended from something like the great apes. And just have a look at this for a moment. On the left, you see a human pelvis and vertebral column and ribs. And here, the middle one is the Rudopithecus, the one that I'm attempting to make at the moment. And Allegra over there is currently making my human pelvis. Can you hold it up for a moment? And you can see she's just applying the very first coating of paper mache to the armature. I'll talk you through the armature and the paper macheing soon. Right, thank you. As you see, I'm employing child labour today, which is entirely unethical. So, the other one here is an orangutan skeleton. And you can see a few differences. If humans descended from the orangutan, there's this issue about these huge iliac outgrowths at the top, and they, of course, are around much of the lumbar spine, and they would reduce its flexibility, and they would make it quite difficult for this organism to stand upright. And of course, orangutans don't. And here we've got the difference. We've got the small iliac outgrowths, and they don't enfold the lumbar spine so much in a similar way to humans. You can see there's a great deal of development between the Rudopithecus and the humans, and the Rudopithecus has 1, 2, 3 lumbar vertebrae before the ribs start growing, and the human has 1, 2, 3, 4. So there's quite a difference in the spine, 1 whole extra vertebrae. So firstly, the methods of the actual sculpture, and you probably won't be doing that at the moment, but I am imagining that we'll get back to school at some stage this year, and to me this project is still very active. So, last time you saw this, and the biology room is still littered with the chicken wire skeletons of your sculptures, but many of you were starting to cover them with a bit of tape, and you can see what's happened here with the tape. It's quite a good way of being able to sculpt and mould the chicken wire frame. When you're doing the chicken wire, do use gloves, because you will lacerate yourself with the chicken wire ends, and they're not very forgiving when they go through your skin. So yeah, I used these, then I coated it with the tape, and the tape's excellent because you can mould it now as if it's made of clay, and it makes it a very, very different sort of medium than just chicken wire. And so what I've done is moulded this, and here's the 3 parts of the pelvis: the ilium, the pubis, and the ischium. And here we've got the very important parts where the sockets are, where the femur is joined on. Alright, so that's my basic armature here. And as you'll notice, Allegra's doing quite a lot with the paper mache paste, and you'll notice it's quite runny. You can buy a paper mache paste at Michael's or Ben Franklin's or somewhere like that, but don't. Just make it yourself. It's made of flour and water, and you just mix them until you get something like about that consistency. And then add quite a lot of salt. And the reason the salt's there...
Pancake batter.
Like pancake batter, yeah. The reason the salt's there, it tastes very salty, is that it prevents rotting. So you don't want your paper mache sculpture rotting, and so you don't want bacteria attacking it. And so if you've got lots of salt there, if bacteria do go in, all the water will leave them by osmosis, and you'll get dead bacteria, and they won't be able to do any decomposition of your valuable sculpture. So you can see that process going on, and that will continue as we talk through the video. So now I'd like you to have a look at what you need to do and I'll go through it using mine as an example because you can't really talk about it academically, you have to use an example. Now the cover, as usual with me, the first bit comes last, so I won't talk about that. So the general effect of a mutation on proteins is simply the mutation is a sudden, fairly random change of some of the A, G, T, C bases in DNA. And because those in triplets code for the amino acids that make up the eventual protein, then they'll make a difference in the amino acid sequence. And because there's a difference in the amino acid sequence, the one that was there before is now replaced with one with different properties. So it might be hydrophobic instead of hydrophilic, for example, and therefore the amino acid may produce an entirely different shape in the protein. And that means perhaps that the active site that was there before is no longer there, or perhaps it creates a new active site and an enzyme is able to work on this new protein, whereas it couldn't on the last one. And it might alter things quite radically. So the function, the original shape, here's my original Rudopithecus pelvis shape. It's long. You can see here's the coccyx, and here's where the spine, the vertebral column, would attach to it. And it's long, and the length of it means it's quite inflexible. The human pelvis, can you hold that up for a moment, please? Sorry about that. You notice it's much, over this way a bit. It's much shallower, and it's broader. And here's, of course, the hole where babies can come through if there's birth there. Here's the ischium, here's the pubis, and here's the ilium. And you'll notice that being a lot shorter allows a great deal more flexibility, which allows us to stand upright, whereas these poor things were condemned to be at this sort of angle. Now, thank you. This sort of creature, it's thought, didn't brachiate through the trees, like hang from them and swing along. So you've probably seen gibbons or orangutans doing that at a zoo or on nature programs. This one appears not to have been like that. This tended to walk along branches, and as you've seen monkeys walk along branches sort of horizontally, keeping their back parallel to the ground, these ones were a lot more upright, and that gave them the freedom to grab things a lot more easily than perhaps monkeys who were horizontal did. And so here's the idea of the change in it. The function of the original shape, it walks along like that, which enabled it to walk along branches. And the change was the human one, that allowed it to move onto the ground completely. So it adopted a completely upright posture and probably adapted it to a very changing environment. The thought is that much of Africa went from a forested area to a savanna grassland area. Okay, now let's get on with what we're doing now, the 5, 6, 7, 8. So the gene affected, the protein modified. Now, most of you, I think, when I'm looking at your projects, are doing something about limbs, so something about horses' hoofs or turtle flippers, or I've forgotten the other ones, but you've got issues where it's changes in shape, size, or function of the limbs. And these are due to the Hox gene. So the Hox gene complex, the gene loci on the human genome now, where there's a bunch of genes which control the body pattern, the bilateral symmetry and the development of 4 limbs and the usual sort of 5-digit sort of things that we're used to in so many of these organisms. And they're largely controlled by these things called Hox genes. Now mine goes a bit further. I'm looking at the PIT-X1 gene. And there's a PIT-X1 and a PIT-X2, and I'm not entirely sure what the difference in function is. But what they seem to do is they encode a transcription factor, and that transcription factor unlocks the bone development in embryonic times. And as with most of you, you'll be looking at differences that happen in embryology, because by the time the organism is born, that gene has already been expressed, and it's the body shape that it will pretty well have for the rest of its life. But during embryology, of course, during the embryo phase, it goes through the time of having its gills, having its tail, the tail disappearing, forming the limb buds, which then grow into the appropriate sorts of shapes of the adult organism. And this gene, this PIT-X1, what it does is it has this transcription factor which allows the RNA transcriptase to attach to the beginning, the initiator site, of a gene that codes for pituitary hormones, such as prolactin, which causes milk development, but the one we're kind of interested in is pituitary hormone, which acts as human growth hormone. And this one, of course, depending on the time during the embryonic development at which it is expressed, can cause different bones to be different lengths. And for example, when this area of the ilium of this Rudopithecus was developing in the in its embryonic times, then presumably there was quite a lot of pituitary hormone going on, being expressed and allowing this to become longer than it is currently in the human one. So that's my thinking here with what my gene is. Now, we go on now to the Darwinian stuff. So what the mutation is and how that affects variation. And that's really, really simple. So the mutation is the PIT1 gene, the PIT1X gene is changed from the Rudopithecus to the human, and what appears to happen is that it's now transcribed at different times and at different lengths of time during the embryonic phase. And that means it's causing these differences. And here's the variation. The variation is this and this shallower one. So presumably, at some time of human or pre-human evolution, there was a stage when there were things wandering about with pelvises a bit like this, meaning that they walked sort of on their forelegs, on all four legs in trees. And also at some stage there were these shallower pelvises that were pretty cumbersome when the pre-humans were wandering about in trees, but were a lot more useful when they were walking about on savanna grasslands. So there were those two variations, the long pelvis walking like this, and the short pelvis walking upright so you can't see my head anymore. And here's the next thing. Variation, of course, gives the raw material for natural selection. Natural selection acts on variation because there's two different kinds of variations, there's two very different kinds of organisms wandering about. Then natural selection can start to determine which one is more likely to survive and reproduce and which one is more likely to die out before providing its genes to the next generation. And then we get overproduction. So with overproduction, there's that thing that Darwin sort of realized, that all organisms produce many more offspring than actually survive to reproduce. And that means, in this context, when we're looking at the development of the Rudopithecus type long pelvis to the human type short pelvis, we've got the idea of, well, this wasn't naturally selected because it hasn't survived. The human shorter pelvis has survived. And so presumably what happened was overproduction, not enough resources for all of them to survive and reproduce, competition for resources, and what happened was these tended to lose out. These tended to get a lower helping of the available resources, and maybe they didn't tend to live as long and therefore reproduce as much as the ones that got more resources, which was now the ones wandering about on the grassland, being able to look up and see where the resources were, being able to use their hands to trap and capture the meat that they would eat then. And so we've got the overproduction. The natural selection selected these ones not to survive in as great numbers, these ones, the human type, to survive in greater numbers. Now we get to the last three, and the first one you're responsible for very soon. So how selection pressures acted on variation. I think I've kind of done that, but now I want to link that to changes in allele frequencies. So I've got my PIT1X gene I'm talking about. There's two alleles for that. One that leads to the pituitary hormones being expressed at a time that would make this kind of pelvis, and the other variation with the pituitary hormones being expressed at a time that would make the shallower human type pelvis, which I see the first coat is nearly finished. I'm quite happy about that. So the change in allele frequencies. So we've got the idea that these ones don't tend to reproduce as much. The new human type tend to reproduce more. And so, going back to the Hardy-Weinberg idea, this is not a population in Hardy-Weinberg equilibrium. There's natural selection working. And so, instead of the frequencies of the two versions of the PIT1X gene staying the same or roughly the same from generation to generation, here we're going to get a situation where the Rudopithecus type PIT1X gene decreases as the generations go on, and the human variation of it increases as the generations go on. It's a pretty simple concept, but you do have to express it in the right terminology. And the very last thing is the resulting adaptation. And of course, we've talked about that. The resulting adaptation is no longer a long pelvised organism that walked horizontally on branches. Now it tends to be an organism that has an upright stance due to this much wider, shorter pelvis. Now, when you do this, I suppose you'd be thinking about what you're going to put on the cover. It's completely up to you, except what I ask for is that the cover is eye-catching. And it sort of reflects what you've got going on in here. I've got 4 criteria. I've forgotten what they are, but they're in the rubric. Okay, so I imagine right now you're not going to be constructing your models. We'll do that if and when we get back to school. But please do work on your booklets, because obviously they all go together very soon, and the final copy is due in not much time at all. Okay, we're going to keep making our pelvises, and well, good luck to you.